Great, thank you. Great to be here, and I want to thank the organizers for putting this seminar together. So I'm going to talk a little bit about work we've been doing over the years on modeling drift diffusion dynamics of proteins within lipid bilayer membranes. I think in this audience, don't need to say too much about the background biology, but I'll just kind of point out some of the most salient points for our work, and that is, membranes are these sort of very dynamic, rich structures. Obviously, proteins are patterned and play many interesting roles within lipid bilayer membranes. And the real, I think, complexity in understanding the cell biology of membrane protein function is really this interplay, not only between the individual proteins, but also how they interact with sort of the intrinsic physics of these soft materials. And so, as you can already see in the upper left there, membrane exhibit all kinds of really interesting geometries. These are dynamic structures that are constantly undergoing shape changes, topological changes, and all of this is sort of happening at the same time that all kinds of interesting metabolic activity and other types of mechanical functions are occurring with proteins that are basically anchored and organized within the bilayer. So lots of rich, I think, physics and opportunities there, both for modeling and for interesting experiments. And so what I'm just kind of showing here on the upper right is some work where there's molecules that are embedded within a lipid bilayer that, when activated by a light source, will change the local preferred curvature. And already you can see the sort of local preferred curvature manifests in these sort of global interesting morphological changes. And then on the lower right is some experimental work of a colleague we were collaborating with, where they're doing single particle tracking of proteins, and you see really interesting differences in the manifested in the diffusivity depending on where the proteins are in the geometric structure. And so I'm not sure if it's showing up here on Zoom, but there's like this little nanotube that's been pulled out of a aspirated vesicle, and you get different diffusivities if you're kind of in the nanotube due to the dissipative mechanisms that are most relevant there versus in the bulk vesicle, as well as interesting dynamics and structuring that can occur at these kind of junctions. So again, interplay between potentially hydrodynamic effects, and I don't mean necessarily flows per se, but the local diffusivity arising from hydrodynamic coupling affecting collective dynamics, the elasticity of the membrane, obviously, as you can see, geometry. And then, of course, on top of all of that, in cell biology, we're dealing with systems that are typically out of thermodynamic equilibrium, right? There's energy exchanges given the hydrolysis of ATP, GDP, other chemical processes. So we'd like to develop some biophysical models that can incorporate some of these effects when we're studying the interactions of membrane proteins. So that's kind of motivation for the work. So I'll talk a little bit about how we're going to model the hydrodynamics. And again, if you've done a lot of work in this field, you'll be familiar with this theory. If not, this is worth knowing about as some background. So if you even want to look at the passive diffusivity of a single protein within the bilayer, you already run into some interesting issues, which is if you look at the viscosity of a lipid bilayer, it's typically on the order of like 100 times more than that of the surrounding fluid. And so you might be tempted to model that as a pure two-dimensional sheet, but you immediately, if you go through the analysis, realize there's a problem here, because if I put a particle in a purely two-dimensional sheet and I push on it, there's no steady state velocity. And so that seems a little counterintuitive, but what's really going on here is if you're modeling it using the usual Navier-Stokes equations, two-dimensional sheet, it's as if you're assuming like a symmetry in the z direction, and it would be as if you were pushing on an infinitely long cylinder through the fluid. And so you actually get these logarithmic divergence and things. Obviously, in the physical system, the system is never truly infinite, so it means there's got to be some additional regularizing length scale or physical effect that's being neglected in the modeling. And so the way that that's going to be dealt with, and this was known by Safman and Delbert, because if you kind of look at the membrane, you'll start to get that sort of long-range flow structure, but then what will happen is there's traction stresses with the surrounding bulk fluid, and that actually serves to regularize things and give you a finite velocity. So the sort of takeaway here is, even though the membrane viscosity is very large, we can't ignore the bulk three-dimensional flow. So if we take that into account, you can actually work out an asymptotic theory for what the mobility responses will be, and you get a number of really interesting effects. So you work out the asymptotics as the slab thickness goes to zero, and you find things like this logarithmic expression for the mobility response. And I'll point out a number of the interesting features for our purposes, which is first off, you notice it has a logarithmic dependence on the particle size. So that's quite interesting because in the bulk three-dimensional fluid, it tends to go 1/the inverse of the particle size. And so that means you're going to have a very weak dependence on the particle size. And there's also this important length scale that comes in, which is referred to as the Safman-Delbruck length scale, which is the ratio of the viscosities. And that's kind of where that traction stress effect is coming into effect. And if you do a back of the envelope calculation, you realize that that's on the order of microns. So even though the proteins are very small, and based on your three-dimensional bulk fluid mechanics, you might think everything's going to be very localized, there's actually going to be significant long-range coupling because of this sort of two-dimensional nature of the fluid film and the large viscosity. So many, membranes, if you just kind of look on the length scale of microns, are going to exhibit significant heterogeneity. There's going to be potentially curvature, even potentially topological effects that play an important role that need to get taken into account. Um, so the question is sort of how can we take something like the Safman-Delbruck theory and start to move it towards these more realistic and I would say rich scenarios that arise in cell biophysics, similar to what you're seeing out there on the lower right. So I'm going to start off kind of as I zoomed in there. I'm going to start off looking at a single vesicle, and then we'll look at larger scales. And so if you wanted to model a single vesicle, one way you could do that is basically take that sort of Safman-Delbruck idea, model it using fluid slabs for the two leaflets, and then we have a bulk interior three-dimensional fluid and a bulk exterior fluid, intermonolayer slip, etc. Already, I think there's some rich things to do there to study liposomes and other small vesicles. I'm not going to go into all the equations here on the left. It's a little bit much to explain, but pretty much by using tools from differential geometry, we can extend all of those hydrodynamic equations that I showed or mentioned earlier to the curve setting. And the real takeaway here is you can generalize the Laplacian, and you get things, contributions from the local Gaussian curvature and other effects. So there's interplay between the flow field and the geometric structure. That's really the key point there. And these are the gory details of how you do the bookkeeping for that. Then you'd have the bulk three-dimensional fluid surrounding the vesicle that's given here, traction stresses kind of coupling everything together. Okay, so in order to model proteins now, this is basically the background hydrodynamic theory, we're going to take an immersed boundary approach where the idea is we don't want to just model the proteins as some continuous concentration field because we're really interested in the detailed kinematics and kinetics of interactions and also the patterning, kind of as I've shown previously. And so we have a model that allows you to simulate for proteins that either span both leaflets or in one or the other leaflet. And the idea is we're going to treat those as effectively markers in the fluid. So this will give us a discrete particle model. And the way that we're going to apply any force that acts to a particle, a protein in particular, to the membrane is through some force spreading operator that I'm kind of showing here with this little blurred out Dirac delta. And then if we want to know the kinematics of the protein in response to the other proteins, motions, basically through the hydrodynamics, then we do a local averaging using that same kernel. And if we put all that together, that's got some really nice properties, and you can show that it will recover much more detailed models that you might construct for the hydrodynamic coupling. And so we recover things like in the bulk three-dimensional fluid, the Oseen tensor, we would recover the Safman-Delbruck theory, and then in near term, you get a regularization known as the Rotney-Proudman-Yamakawa coupling. So that works quite nicely, and we've extended that to the membrane setting for both particles and translational motions of particles and for rotational motions. So obviously, when you have particles that come together and dock, it's quite important also to have the rotational degrees of freedom potentially taken into account. And so you can both get the flow responses for forces as well as torques acting on things embedded in the membrane. And so we've developed a solver for that. I'm going to kind of skip over this in the interest of time. There's some interesting things that come out of the analysis of the fluid mechanics as you're developing the spectral representation, as I'm showing here. But this allows you to then simulate the collective dynamics of potentially a very large number of proteins in the membrane. I think I'm going to skip over some of the interesting hydrodynamic phenomena that we found doing these calculations on vesicles. There's been a fair amount of experimental work trying to estimate membrane viscosity from observations, and that's kind of a tricky thing to do because, as I showed before, the logarithmic dependence on the particle size means that Reading out diffusivities can be quite challenging if you try to do that for different protein sizes, for example. Another approach has been to look at sort of bulk flows that induce basically rotational fields in the membrane. And so you can use things like R theory to predict where that recirculation vortex would be as a potential estimator for the membrane viscosity. And so that's what I'm just showing here is kind of a chart of how the ratio of the Safman-Delbert length to the radius of the vesicle can be used to basically predict where this vortex center will be. And you could use that as a proxy to get the viscosity estimate. You can do other interesting things and look at how proteins behave. For example, if I were to push on a protein on the outer leaflet, how would it respond? This has implications for how the passive diffusivity would work because basically the mobility responses through the Stokes-Einstein's relations are the collective diffusion coefficients that, a tensor that you would have for a collection of proteins that are interacting with one another. And so I won't go into all these detailed studies. I'll just kind of refer you to our 2016 paper on that, but you can get really interesting and subtle correlation and anti-correlation effects due to the hydrodynamic coupling, which could be quite important in different chemical processes occurring within the bilayer. Um, so I think yeah, and so what I want to talk about is if you're going to do a large number of proteins, kind of like I showed before, it makes sense to go beyond just computing mobilities and to look at a background fluctuating field. And so we have ways of extending these ideas to basically be able to do simulations of large number of proteins that are patterned in the membrane, but now where the source of the fluctuations is modeled through a fluctuating hydrodynamic field. And so I'm showing the inertial representation here on the left. Obviously, in the cell biology context, a lot of this would become overdamped, and so you can take asymptotic limits of this. Really, the key salient point is it's the fluid equations I showed before, plus this drag term, which is going to model the interactions between the protein and the surrounding fluid. And so just get my laser pointer here, that's right there. And so you have an equal and opposite force in the protein momentum equations, and then you can thermostat all of this using principles from statistical mechanics. And so again, I won't go into a lot of the details, but refer you to our 2019 paper if you're interested in how you derive these kind of fluctuating hydrodynamic descriptions for these curved surfaces. So we refer to this as surface fluctuating hydrodynamics. And then if you're interested primarily in the overdamped regime, you can, take a limit of this, and there's ways to use this background field to compute efficiently the coupling, hydrodynamic coupling tensors. And so now you would have your collective protein dynamics with the appropriate hydrodynamic correlations that impact phenomena occurring in the membrane. And I'll show some examples where the hydrodynamics has some impact. Even if you look at something like the pair meeting time, if you model it with Langevin dynamics, you get this kind of very spiked and localized time scale for two particles coming into contact. If you have hydrodynamics, you tend to get these very long tails. So it does have a quantitative impact on things as simple as pair meeting times, which are obviously very important if you had something self-assembling or other types of chemical reactions occurring within the membrane. And you can get interesting interplay between the hydrodynamic effects and the chemistry. And so what I'm showing here is a mechanism by which we have little transient clusters that form in the bilayer where particles can switch back and forth between being attractive or repulsive. And there's a time scale governed by the switching rate, lambda. And you can kind of see in the case where lambda inverse is very small, there'll be no clusters. And when lambda inverse is very large, you'll kind of get bulk clusters. And you can look at what happens with and without hydrodynamics. And given, again, the impact that has on the kinetics, for Langevin, you pretty much get exactly the same max cluster size. Whereas for hydrodynamics, you get some really interesting and subtle effects based on the time scale for the particles to come together versus the switching time that impacts the max cluster size, as you're kind of seeing in this little experiment. So there's potentially interesting ways that you could come up with mechanisms to regulate based on the biophysics of these hydrodynamic correlations is what we're getting at. And so there's also things that I could say about the velocity order correlations that also manifest that depend also not just on the hydrodynamics itself, but also on the ratio of that Safman-Delbert length scale to the size of the entity that you're modeling, whether it be some kind of organelle membrane or things like vesicles. Um, And so you can do things as well looking at active mixing. So obviously proteins aren't passive within bilayers. And so this is just showing a little experiment where we have a Golostanian swimmer, but you could imagine just proteins that are undergoing conformational changes. That's going to push the lipid around in the surrounding solvent. And so this is quite interesting because given all of the protein inclusions, you could imagine that the bilayer membrane would have very high viscosity. And so if you're just waiting for passive diffusion to mix things, that may be something that takes a very, very long time scale. But if you account for things like the active conformational changes or even active transport that's occurring, nearby cytoskeleton, you could potentially end up with these hydrodynamic effects resulting in mixing of the proteins and getting much more effectively enhanced diffusivity. So that would again be a mechanism by which, by turning on and off these active conformational changes, you could be tuning up or down these important kinetic parameters to turn on and off different biochemical or cellular processes. And so here I'm just showing a little plot of this study on the left where, again, it's kind of a toy model, but it kind of illustrates the idea of having a Golostanian swimmer. Basically, as we have the number of swimmers increase, you can see the enhancement in the effective diffusivity of these additional passive proteins. So interesting potential there, I think, for some biophysical mechanisms to, again, kind of upregulate or downregulate, different chemical processes within the membrane through active processes. This just gives a hint of some of the other things you can do with that. You can look obviously at the actin cortex and other types of polymeric structures. You can think about spectrum networks and red blood cells, et cetera. All of that can be modeled pretty readily in the framework that we've presented here for surface fluctuating hydrodynamics. I'll kind of refer you to the papers down here for people interested in that work. This 2016 is a good entry point where we go through a lot of the initial formulations, and then these are some of our more recent studies. In the last bit of time, I'm just going to talk a little bit about recent work we've been doing on extending a lot of these ideas to the non-equilibrium setting. And so there's been a lot of interesting experiments using basically optical traps to move proteins around, but there's been more recent work where they use these thermal traps, basically lasers that heat up part of the membrane to push particles around or to ablate or change part of the membrane structure. And so this is just showing a little experiment where if you have a little laser trap that is sufficiently strong, this can act to repel particles through this thermal phoresis type effect. Or similarly, you can use it to ablate or basically remove proteins from certain parts of the membrane, turn off the laser, kind of look how things recover. And of course, in cell biology, there's lots of interesting instances of thermal gradient sensing. There's experiments in neuroscience where you have dendrites that grow towards the heat gradient. You have obviously bacteria that need to sense gradients to move towards or away from sources of undesirable temperature. And so what we've done is reformulated a lot of what I showed you previously using some tools from non-equilibrium, statistical mechanics. And I'm not going to go through all of the gory details, just to say that we can basically take that model that I showed you a moment ago with fluctuating hydrodynamics, and we can kind of extend it by tracking in addition thermal fields. It does get a bit challenging how you do all the bookkeeping because you need to appropriately account for the energy exchanges. And so what we've done is basically work that out in the inertial regime, and then you can take asymptotic limits to get the overdamped results. And so what's really interesting here is you can have particles that are heated up by some external source, and then you can track how the heat changes in the membrane, and of course, how that all feeds back on the drift diffusion dynamics. And so you can do things like hot Brownian motion, where particles move, affect the membrane, and that sort of feeds back in actually impacting the diffusivity. And there's been some interesting recent experiments on that type of phenomena. More, I would say, for in vitro kind of biophysical type probing of different parts of the soft material properties of membranes. So this is the particle temperature. We found in our formulation it's useful to have a little interfacial region. You can think about it as the particle plus the lipids nearby as an additional degree of freedom, and then we have the membrane flow temperature field. And that's what's being shown here. And then you have interesting energy exchanges. I'm kind of accounting for everything. A lot of these terms would disappear because things like CF might be very large for certain specific heats. or some of these frictions might be relatively negligible, and there's ways of taking those out. And so this is how the mechanical energy is getting kind of eaten up by the fluid viscosity. This kind of heats up the fluid, obviously. This is some of the mechanisms by which the exchange occurs through Fourier's law, effectively, between the particle and the interfacial regions. And then, of course, you have lateral Fourier transfer of heat, within the membrane itself. And so there's a lot of potentially interesting things you can do with that. I'm going to skip a lot of the numerical details that gets a little involved, but we've kind of worked through how to do the sampling and
You have about 3 to 4 minutes.
Okay, all right, we'll do. Yeah, I'm going to wrap up soon. Okay, so this is just showing a little fluctuating hydrodynamic experiment on the right where the particle's pushed, heats up the medium, and you can see how that kind of feeds back a bit on both the diffusivity and the motion of the particles. You can also have externally heated particles, and then this is just showing the fluctuating temperature fields in the membrane, as that particle kind of diffuses around. And then through the heating up of the particle, if you had an external source, the diffusivity as well would increase. In this case, the particle was just kept at a constant temperature, and so you're just seeing the result on the surrounding membrane. Okay. Alarm there. And so anyway, there's very interesting things I think one can do with this, and I'd be very happy to talk with people about experiments or other biophysical projects where these types of techniques might be of use. I'll just give a few very quick initial experiments we've been doing, or simulation experiments we've been doing with these techniques. One is looking at protein localization, right? Obviously, membranes are patterned in highly structured ways, probably from an interesting interplay between anchoring sites from the cytoskeleton and biochemical mechanisms. And so this is just showing, again, the subtleties that come up when you start taking into account the background biophysics. Here, if the diffusivity of that chemical field was pretty small, the protein would just move to where the high concentration signaling molecule is. And we're assuming an attraction between the signaling molecule and the protein. Whereas if the protein is unable to move very quickly, maybe it has a large drag or the diffusivity is fairly large of the signaling molecule, that will tend to get attracted to the location of the particle. And so we're looking at this trade-off using these fluctuating hydrodynamic techniques, and you can kind of see there's an interesting interplay between those two timescales of whether the particle moves to where the signaling molecules are, or the signaling molecules move to where the particles are. And again, these are little kinetic parameters that you could imagine through biochemical means you could tune in order to help pattern proteins in different locations within the membrane structure. So we have some preliminary results on that. This is some thermal gradient sensing in the presence of fluctuations. So where you have a very weak gradient, how might you encode that biochemically? There's interesting trade-offs in kind of your spatial temporal resolution versus filtering out noise. And so we have some simulations that we're doing to sort some of that out. And more recently, we've been also looking at these optical trap experiments where there's an energy landscape that the proteins diffuse. And if you heat the particle up, how does that impact escape from different energy wells within the landscape? And so you could imagine this being a way to probe what the energy looks like within these crowded protein bilayer structures. And so some interesting results here as you kind of change the laser strength and the particle heats up, affecting its own Brownian motion, so hence hot Brownian motion, how that affects the effective kinetics within these complex energy landscapes. So I'll kind of refer you to the papers for more details on this work. And like I was saying before, we'd be very happy to talk with people about potential collaborations if there's things going on in your labs where some of these simulation techniques might be of interest, or if there's also other biophysical questions that, that might overlap with some of this. So thanks everybody for your attention. Really great to be here and discussing some of these topics with you. Thank you.
Well, let's thank Paul for his great talk. I see a question in the chat from Robin. So did you encounter protein-protein attraction mediated by the membrane under equilibrium conditions?
Yeah, so we have other work that I didn't report on today where the proteins can bend the membrane. And in that case, you can get attractions and repulsions that are very long range from the mechanics of the membrane. But here, for now, the membranes are kind of static geometry. Yeah, so that's a good question.
Can I ask a second question?
Sure.
For that kind of proteins which you just mentioned, for the case of viral assembly, you have proteins which are attracted by positive Gauss curvature, and they trigger kind of budding, and then proteins with negative Gauss curvature attraction are then attracted to the negative curvature
Hmm.
of the neck and pinch it off. Is that a
Hmm.
possibility for you to study that?
Yeah, so you could do it both for static geometries or potentially changing geometry. And there's been some work on coarse grain models. I'm sure you're aware of way back quite a few years now. But yeah, so in theory, we could do that quite readily for the static case. Yeah, because you would have the curvature fields entering somehow into your effective free
Exactly.
energy
Yeah.
for the proteins.
Yeah.
Yeah, yeah, yeah. That could all be simulated. Yeah.
The negative Gauss curvature proteins are active. They actively pinch it off, and it is not clear if that could be done under under equilibrium conditions.
I see. You're saying somehow the active pinching is changing the local temperature, or
It's just I don't know details. It's called the escort machinery. These are laminar-like proteins. They are attracted to negative curvature, and they apply such a large force that it causes it causes formation of a vesicle that pinches off. It's also occurring under natural exo and endocytosis processes.
I see, I see. Yeah, so you're saying it's such a large energy because you're getting a change in the topology of the structure and all these little
Exactly.
things, and you have to overcome that energy barrier of all that detailed lipid organization
And it's
somehow pinching
impossible
the
to model
structure.
that within continuum theory.
Yeah, yeah, I agree. You would need some multi-scale type model or a coarse grain model. Yeah. Oh, those are very interesting things to think about. Yeah, so our framework so far in this part of the work is all continuum based.
So there's another question from Yijian in the chat. Uh, so I imagine the theoretical and numerical framework would become very tricky if we allow the membrane to be flexible.
Yes, yeah, that is a very fair question. It's possible, but then the problem is, see, every time you introduce a new physical phenomena, you're introducing new timescales, and so some of these timescales become very rapid. Like the undulating motion of the membrane, and so you really kind of want to be judicious about which effects you include and which ones you neglect. And I would argue, even with the frameworks I presented today, what you'd want to do is start with those as baseline, some of those more comprehensive models, and then asymptotically remove pieces of it depending on the particular problem, because yeah, it can get quite computationally involved. So we know how to do that formulation. We'd probably do it more from a variational perspective as opposed to writing PDs down. But yeah, it does get numerically quite involved, and yeah, we've done a little bit of work on that. But you are correct that that's part of the reason why we're considering primarily static structures, is to avoid some of those challenging geometric and temporal stiffness issues. Yeah.
Um, so maybe I have a quick question about one of the earlier results that you show, like when uh, you consider two particles like diffusing and
Yes.
colliding into each other on like a vesicle, and so the time distribution is much, much wider than the non-hydrodynamic models. And so, like, how do
Yeah, I
you
can find
sort
it
of
here. Yeah.
intuitively, yeah, it's one of your very early
Yeah, yeah, okay. I don't
example.
think I won't attempt that. Yeah.
So am I understanding correctly that it's because of the hydrodynamic coupling, and so when one particle is moving, it's kind of pushing the other away in a sense?
Yeah, yeah. So there's well-known effects with colloids of like where you have lubrication effects where it's like they almost never will completely touch, right?
Mm.
And so it's similar to that. It's like as you come together, you have to push all this fluid out. So that really impacts the probability distribution for those kinetics. Yeah.
Okay.
Things kind of
Thank
slow down. Yeah. I mean, I'm not sure how to explain it in a very simple way, but yeah, it's because of the hydrodynamic flows and the fact that you need to push fluid out in order to come into contact that's leading to those long tails.